progress began to stall around the 6th month. The same 30-minute walk that once left you lightly out of breath now barely raises your heart rate. Your muscles need more time to recover than they did at 40. A session that once required a single rest day now demands 3 or 4 before the soreness fades. Sleep becomes lighter, easily broken by small sounds that your younger self would have ignored. Your skin heals more slowly, with minor cuts and scrapes lingering for weeks instead of closing within days. Your ability to focus shortens, with a deep, steady concentration you had in your 30s starting to fade by mid-afternoon, and the energy that once carried you smoothly from morning to night now dips around 3 o'clock, leaving you reaching for coffee you never needed before just to finish the day. Each of these appears to be a separate issue: a fitness plateau, age-related muscle changes, sleep disturbance, slower skin repair, reduced focus, and declining energy. 6 different concerns, each with its own label and explanation, the kind a doctor might address in separate appointments, treating one system at a time with no overlap between them. Yet beneath all these symptoms lies a shared factor. There is one single molecule that every mitochondrion in every cell of your body depends on. It converts food into usable energy, helps repair damaged DNA, regulates when genes turn on and off, and supports the countless chemical reactions that keep your systems functioning. Instead of 6 separate causes, there is one molecule underlying all of them, and it has been gradually decreasing in your body since about the age of 40. By the time you reach 60, measured levels show it at roughly half of its peak value. That kind of decline changes how you understand what is happening inside your body. It is real, measurable, and steadily worsening year after year. It affects all these systems at once for a simple reason. They all rely on the same limited supply. 6 issues on the surface, but one shrinking resource beneath them. Imagine a large building with 6 departments, each on its own floor, all powered by a single generator in the basement. For years, that generator runs at full capacity, supplying more than enough power to every floor. Then gradually, its output begins to drop, falling toward half of what it once produced. The upper floors start to dim. Machines slow down on one level. Computers reset on another. Each department assumes the problem lies within its own wiring. They call technicians, inspect circuits, and attempt isolated fixes. 6 floors, 6 sets of repairs, all focus in the wrong place. Because the issue is not in the wiring at all. The real problem is the generator itself, producing less power for the entire building. Your body functions in much the same way. The molecule is that generator. Yet very few people, whether patients or doctors, are familiar with its name. So let's identify it, understand how it fuels every system, and explore why your body gradually loses the ability to maintain it. The molecule is called nicotinamide adenine dinucleotide, a name so complex that is usually shortened to NAD, often written with a plus sign to indicate its active form. Think of it simply as NAD. It is a coenzyme, meaning a small but essential helper molecule that allows other biological machinery to operate. It exists in every cell of your body, from brain neurons to gut lining to heart muscle fibers. No cell can function without it. Its most basic and essential role is to transport electrons, a process that may sound abstract but is critical to life. Every action your body performs, every heartbeat, every thought, every movement, depends on a molecule called ADP, the primary unit of energy in living cells. ADP is the currency your body spends to carry out all functions, and it depletes rapidly, within about 20 seconds if blood flow stops, which is why loss of circulation leads to immediate collapse. When ADP runs out, the body can no longer sustain consciousness or activity. Mitochondria, the small power stations inside each of your cells, produce ADP through a process known as the electron transport chain. In this system, NAD acts as a carrier that delivers the raw material needed to start the process. It collects electrons from the food your body has broken down, transports them to the chain, hands them over for energy production, and immediately returns to repeat the cycle, performing this task millions of times every second in every cell. It functions like a tireless courier. Remove NAD, and the entire chain is left without fuel. The assembly line halts, ADP production drops, and a cell lacking ADP cannot maintain its structure, produce proteins, repair DNA, or send signals. Without sufficient NAD, the cell begins to fail quietly, starting with its energy supply. In this sense, NAD keeps the energy system running. However, it also serves a second, more unusual role. In this role, it is not reused but consumed entirely, disappearing once its task is complete. Two main groups of enzymes are responsible for this consumption. The first group, called sirtuins, act as regulators that interact with your DNA, turning certain genes off while activating others that help protect the cell under stress and reduce inflammation. You can think of them as supervisors maintaining order within the system. Each time a sirtuin performs its function, it uses up one molecule of NAD permanently. The second group consists of repair enzymes known as PARPs, which act as emergency responders for DNA damage. They constantly scan your genetic material for breaks, and when damage is detected, they move quickly to repair it. This process also consumes NAD, often using multiple molecules in sequence to mark the damage and coordinate repair. At the same time that NAD is being used repeatedly to generate energy, it is also being steadily spent on maintaining genetic stability and cellular regulation. It supports both energy production and maintenance processes continuously throughout your life, making it one of the most actively used substances in your body, despite being largely unfamiliar to most people. Given its importance, the key question becomes how much NAD remains in your body today compared to decades earlier. Research indicates that NAD levels decline by about half between the ages of 40 and 60. This means that within just 20 years, the supply that supports every system in your body drops to around 50% of its peak level. This pattern has been consistently observed across multiple studies, making it one of the more reliable findings in aging biology. The gradual decline happens largely because of one enzyme, nicotinamide phosphorybosyltransferase, commonly referred to as NMPT, which functions as a recycling system that rebuilds used NAD into its active form. This enzyme represents the limiting step in maintaining NAD levels, acting as a bottleneck in the process. As the body ages, production of this enzyme decreases, slowing the recycling process and reducing overall supply. At the same time, the demand for NAD increases. Over the years, DNA damage accumulates due to natural wear and environmental exposure, and each instance of damage activates repair systems that consume NAD. As a result, the body faces a double challenge: reduced production due to declining recycling capacity, and increased consumption due to rising repair demands. This creates a widening gap between supply and demand. What begins as a gradual decline becomes more significant by the time you reach your 50s, when the body can no longer fully compensate. All systems that depend on NAD begin to receive less of it. For many individuals, the first noticeable effect appears in physical performance, such as exercise becoming less effective or progress slowing down. While part of this can be explained by the body adapting to repeated activity, another layer beneath it involves the availability of NAD. The electron transport chain, which produces ADP, depends on NAD to deliver electrons to its first stage, known as complex 1. This step acts as the entry point for energy production, and NAD-carrying electrons is what enables the entire process to begin. With less NAD available inside the cell, fewer electrons reach that entry point. The process slows, and each mitochondrion produces less ADP than it once did. The machinery itself may remain intact. It is simply receiving less of the shuttle that supplies it. This means that by 60, the same mitochondria powering your walk may be operating with roughly half the NAD they had available at 40. Energy production per cell declines, and because virtually every tissue depends on NAD, the effect can appear throughout the body at the same time. You notice it in your legs during the walk, in your heart as it moves blood through the body, and in your brain as it struggles to maintain sustained attention. Exercise capacity declines along with it. The point at which your body stops adapting to the same walking routine arrives sooner, because the energetic foundation beneath the activity has quietly weakened. The plateau in your walking progress therefore has another layer to it. The foundation supporting that progress has gradually dropped. Now consider where this shortage may become especially noticeable. The prefrontal cortex, the area of the brain located behind your forehead and heavily involved in attention and decision-making, has a very high energy demand compared with many other tissues. It is essentially a constant consumer of fuel. When energy availability becomes limited, tissues with the greatest demands can be among the first to notice. That makes the fading concentration in the afternoon and the tiredness that appears around 3 o'clock understandable in another way. Your most energy-hungry organ is trying to perform late in the day after cells throughout the body have already been drawing on the same resources since morning. The available supply has been repeatedly used, while the brain continues demanding energy. In this case, reduced energy production is connected to the smaller amount of NAD delivering electrons into complex 1. So, where does the same molecule become important next? For many people, the answer appears during recovery. Muscle growth has its own mechanisms, including the signals that tell cellular machinery to build new proteins after exercise. But repairing and maintaining that muscle afterward is a different task requiring different systems. Removing damaged proteins, maintaining healthy mitochondria, and controlling the inflammatory response triggered by strenuous exercise all involve sirtuins, the same cellular regulators mentioned earlier. Within and around muscle fibers, several sirtuins help coordinate these maintenance processes, and their activity depends on NAD. Think of them as supervisors who need a limited supply of resources every time they carry out an instruction. When NAD levels fall, their activity can become less efficient. Reduced sirtuin activity can mean that post-exercise maintenance takes longer. Damaged proteins may remain for longer before being cleared. Inflammatory signals may persist, and older or damaged mitochondria may remain in the muscle instead of being efficiently removed and replaced. Each delay reflects a maintenance system operating with fewer resources. There's another problem hidden within this one. Because some sirtuins also help maintain the health and function of mitochondria themselves, the very power stations responsible for producing the energy required for recovery. So declining NAD can affect both sides of the process at once. It can weaken the systems responsible for repair while also reducing the quality of the machinery supplying energy for that repair. The person who once recovered from a demanding workout in 48 hours at 30 and now needs several days at 60 may be experiencing part of this broader shift. The basic muscle tissue is still capable of doing the work, but the maintenance systems responsible for restoring it are operating under greater constraints. Recovery can therefore slow as the NAD-dependent regulatory systems have fewer resources available. Energy and repair are important during the day, but what happens to this molecule once night arrives? NAD does not remain at exactly the same concentration throughout a 24-hour period. It rises and falls according to a daily rhythm that is connected to the body's internal clock. At certain times, levels are higher. At others, they decline. The enzyme in AMPT, which helps recycle NAD, is itself influenced by the circadian system, increasing and decreasing its activity according to a daily schedule. As a result, the amount of NAD available inside cells changes across the day. Research has demonstrated this connection between NAD metabolism and the body's circadian rhythm, and this is where an important feedback loop appears. The sirtuins that depend on NAD are also involved in regulating the molecular machinery that keeps the internal clock operating on schedule. They interact with core clock-related genes and help fine-tune the timing system that coordinates daily biological functions. In other words, the clock influences NAD production, while NAD, through sirtuin activity, also influences the clock. The two systems help regulate each other. When NAD levels decline, that relationship can become less stable. Lower NAD can reduce sirtuin activity, which can weaken their influence over the molecular clock, potentially making the timing system less precise. And your sleep depends heavily on that timing system. The onset of deep restorative sleep, the cycling between different stages of sleep, and the nightly processes that help clear metabolic waste in the brain all depend on coordinated signals arriving at the appropriate times. When the internal clock becomes less precise, the structure and timing of sleep can become less stable as well. As night progresses, the edges of sleep can begin to fray. The deeper stages may become shorter, and waking may happen more easily. The lighter, more interrupted sleep that often appears with age could partly reflect a weakening connection between NAD, the sirtuins, and the circadian clock. But this is where the evidence needs to be handled carefully. Each individual part of that pathway has been demonstrated in separate experiments, yet scientists have not established that the entire chain is a primary cause of age-related sleep changes in living humans. It may instead be one contributing factor among several. The feedback loop itself is well supported, but its overall importance in human aging is still being studied. The most accurate way to describe it is as one thread within a much larger picture, rather than a complete explanation for changes in sleep. The clock may become less precise because one of its regulators has less of the molecule it depends on. That gives us three connected systems, all influenced by the same declining resource. The fourth system helps explain why the decline can become increasingly difficult to overcome. Consider the skin and the way its DNA repair machinery responds to damage caused by sunlight and other forms of environmental stress. At the molecular level, one of the first responders to a damaged DNA strand belongs to the same family of repair enzymes discussed earlier. It detects a break and attaches itself to the damaged area. It then begins creating long molecular chains that identify the problem and recruit additional components of the repair system, consuming NAD as the process continues. The more active the repair response becomes, the more NAD it requires. Around age 40, the balance can still be relatively manageable. Damage has accumulated, but the level of NAD may provide enough resources for repair while leaving sufficient amounts for the other functions of the cell. By 60, however, that balance can shift considerably. 2 additional decades of accumulated molecular damage mean more DNA problems. More damage activates more repair machinery, and greater repair activity consumes more NAD. That leaves less available for sirtuins and less available to support mitochondrial functions. The repair bill has grown, but it is still being paid from the same limited account that supports everything else inside the cell. Worse, the account was already declining before repair demand increased, creating pressure from both directions. Supply is falling while demand is rising, and somewhere during midlife those two trends begin to separate more dramatically. This is where research from scientists such as Eric Verdin and Shinichiro Yoshino and their colleagues has helped clarify the broader picture. DNA repair enzymes and sirtuins draw from the same limited NAD pool, which means they can effectively compete for the available supply. When more NAD is consumed by DNA repair, less may remain for other NAD-dependent processes, including gene regulation. Reduced sirtuin activity can influence the regulation of genes involved in inflammation and cellular stress. Increased inflammation can contribute to additional oxidative stress and tissue damage, which can then activate more repair activity and increase NAD consumption again. Look at the pattern closely, and a feedback loop begins to emerge. Each cycle can place additional pressure on the same shrinking resource. The competition is therefore more than an isolated biochemical detail. It may represent part of the machinery through which aging-related changes reinforce one another. A declining supply can make cellular maintenance more difficult, while the increased demands created by cellular damage can further consume that declining supply. At this point, the entire picture begins to change. Initially, it looked like 6 separate systems simply happened to depend on the same molecule. Now we can see that the molecule has multiple consumers drawing from a supply that is already decreasing, and some of the processes consuming it are responding to the very damage associated with aging. The simple list becomes a feedback system. Multiple tissues, one shared resource, several competing demands, and supply that becomes harder to maintain as those demands increase. That brings us to the question that has been waiting underneath everything from the beginning. If NAD is so important, why can your body not simply produce more of it and keep the supply stable? To understand that, you have to look at the recycling system operating inside your cells, because the body is remarkably efficient at recovering components from used NAD. It breaks down the spent molecule, recovers what can be reused, and sends those components back into production through a process known as the salvage pathway. The name is appropriate because it is essentially a continuous cellular recycling operation. Here is how the cycle works. When a sirtuin or DNA repair enzyme consumes NAD, the molecule is broken down and leaves behind a component called nicotinamide. That leftover material becomes a starting point for rebuilding the molecule. The enzyme in AMPT captures the nicotinamide and converts it into an intermediate compound, moving it one stage closer to becoming NAD again. Additional enzymes then process at intermediate and complete the reconstruction, producing a fresh, functional NAD molecule. The cycle has now returned to its starting point. The molecule has effectively been rebuilt from one of its own breakdown products and can immediately return to cellular work, where it will eventually be consumed again in a younger body. This recycling process can operate quickly enough to replenish the supply despite constant demand. That means the overall availability of NAD depends heavily on how efficiently the recycling cycle can operate. And the speed of that cycle is largely determined by its key rate-controlling enzyme, NAMPT. This is where the entire story comes together, because NAMPT is precisely the enzyme that declines with age. As the decades pass, your cells generally produce less of it, although scientists are still working to understand all the reasons behind that reduction, from accumulated cellular stress to changes in signals that control when the enzyme becomes active. But there's another important twist, one that makes a decline increasingly self-reinforcing. Remember that when NAD levels fall, sirtuin activity can decrease, allowing inflammatory pathways that were previously being restrained to become more active. That increase in inflammation can create another source of NAD consumption through enzymes involved in inflammatory signaling and cellular stress. As tissues become more chronically inflamed, NAD demand can increase further. The cycle begins to close in on itself. Lower NAD can contribute to weaker sirtuin activity and altered cellular regulation, while increased inflammation and cellular damage can create additional demand for NAD, placing even more pressure on an already limited supply. The shortage can therefore contribute to conditions that increase the demand for the very molecule that is becoming scarce. Another consumer is effectively drawing from a pool that was already under pressure from existing demands. And because NAMPT helps control the rate at which the salvage pathway rebuilds NAD, a decline in its activity can slow the entire recycling process. The wheel turns more slowly. The raw material has not necessarily disappeared. The broken down components are still available for recycling. What has become less efficient is the machinery responsible for collecting those components and turning them back into usable NAD. As a result, less NAD can be rebuilt over a given period even while the enzymes and cellular processes consuming it continue operating, and in some circumstances may demand even more. The growing difference between a recycling system that is becoming slower and a body that continues to require NAD helps explain why levels can decline with age. And that shortage sits beneath many of the changes you have been noticing. Is the body simply giving up as it grows older? Not exactly. The opposite is closer to the truth. Your body continues working to recycle and maintain NAD throughout the aging process, constantly try to meet demand with a system whose efficiency gradually changes over time. The effort does not suddenly stop. The problem is that eventually the recycling system may no longer be able to fully keep pace with everything drawing from the supply, and this is the part you could never directly feel. It helps explain why those 6 changes can appear as though they have nothing to do with one another. You cannot feel NAD levels dropping inside your cells. The process is silent. There is no specific pain signaling that tells you the coenzyme is becoming scarce, no warning light announcing that the cellular supply is falling, and no organ sends a conscious message reporting its NAD concentration in the way your stomach signals hunger or your skin registers cold. Instead, the molecule can decline gradually over decades without producing one obvious alarm. What you experience are the downstream effects, and each one can arrive separately at different times, carrying its own explanation. Your afternoon energy begins to fade, while recovery from exercise takes longer. Sleep becomes lighter and more easily interrupted. Your skin repairs itself more slowly. Concentration becomes harder to maintain as the day progresses. Low-level inflammatory activity can become more persistent than it once was. 6 different experiences, appearing at different stages and wearing 6 different disguises, can convince you that each represents an independent problem with its own cause. Yet underneath them may be a shared cellular resource that has been declining at the same time. Return to the building analogy. The generator in the basement gradually loses output, while each floor begins experiencing its own problems. The people on every floor call their own technician and search for faults in their own wiring, but no one checks the generator supplying the entire building. The same basic idea applies here. A molecule that most people have never heard of is involved in processes supporting nearly every system that has been changing with age. The walk becomes harder to improve, muscles take longer to recover, sleep becomes lighter, skin changes, concentration becomes less durable, and afternoon energy declines. 6 systems, 6 conversations, and 6 seemingly separate explanations can emerge. Yet one shared molecule sits beneath all of them. NAD, it is required by mitochondrial energy metabolism, used by sirtuins involved in cellular regulation, and consumed by enzymes involved in DNA repair and other stress responses. Human NAD biology changes with age, and research has reported substantial declines across different tissues and stages of life. At the same time, the systems that depend on NAD continue drawing from the available supply. The decline can be measured, and its downstream effects can be experienced, even though the exact contribution of NAD to each individual symptom remains an active area of research. The important connection is not that every change after 40 has one single cause, but that several systems you assume were aging independently share a common biochemical resource. The molecule you never thought to measure may be one of the underlying pieces of the puzzle behind changes you have noticed for years.